All right. Good afternoon, everyone. It is a great pleasure and honor to introduce my colleague and my dear friend, Dr. Randy Kimple. He is an associate professor at the University of Wisconsin, a very successful physician scientist, and a really role model for all of us. His resume I was looking at is 44 pages, so that tells you a lot. He's fully funded investigator on multiple editorial boards. He's a senior editor at the Red Journal. Randy went to Michigan State University. I was looking at his resume. I'm like, maybe that explains his humility and his brilliance for an undergrad with a BS in environmental science and management. And then he stayed for a long time at University of North Carolina, where he did both his MD, his PhD, his intern residency, chief resident, as well as a postdoc in radiosensitization of breast and pancreatic cancer with Dr. Carolyn Sartor, followed by a transition to a postdoc fellowship at the University of Wisconsin with Paul Harari, where he has been since then as assistant professor. Beyond his accomplishments, Randy has been a champion for all of us in the field of radiation oncology who are physician scientists. It's a tough road. And I am here, I'm a statement as a survivor of the grueling road that it is, that with great friendships and really wonderful support and collegiality, you can make it. So without Randy, I probably would have quit. So it's a great honor and a privilege to introduce to you Dr. Randy Kimple, who's going to talk today about radiosensitization or therapeutic resistance to head and neck cancer. Thank you.
Well, thank you, Sana, and thank you everyone for being here. That was a way too kind introduction, and it's my real pleasure to be here today for reasons that go beyond the temperatures up in Wisconsin. For those of you who have been looking, for those of you who have not, this is what the forecast looked like this morning. I checked just now, and it's a balmy 2 degrees Fahrenheit. But when I get back tonight, knock on wood, it'll be negative 24, which is negative 30 Celsius. So it's colder than your minus 20 freezers are tonight, and a wind chill on top of that. Needless to say, the schools are closed yesterday, today, and tomorrow, and probably Thursday. But our clinic is open and treating patients, as my email just said. Our clinic does not close, so we will keep treating cancer patients up there despite the weather. So I want to start by, I know there's a lot of trainees in the audience here. I want to start by sort of acknowledging the fact that this work is by no means all my work. In fact, the first story I'm going to tell is really essentially a story of a collaboration that has developed over the last 6 to 8 years between myself and Derek Wheeler, who's a faculty in the Human Oncology Department, and has led to co-submitted grants, co-funded grants, clinical trials, and multiple publications and graduate students, and has really been a great example of collaborative science at its best, where each individual brings their area of expertise to the problem. But that collaboration goes beyond just Derek and I to include a pathologist and people in biostatistics, and more recently, I'll tell you a little bit, some very brief snippets about work we're doing with 2 different faculty members in the School of Pharmacy. And all of this has really been, all the data that I'm going to show today has been really made possible through the support of Paul Harari, who's the chair of our department and has led the effort at Wisconsin to bring a head and neck cancer spore grant to Wisconsin, which really has helped to catalyze and accelerate the head and neck cancer research that's going on there. Briefly, my disclosures, none of which are particularly relevant to the talk today other than the funding sources, which I'll mention again at the end. So I'm focusing today on telling you about therapeutic resistance in head and neck cancer. And we've focused on head and neck cancer. I've built my lab on studying head and neck cancer. It's what I treat in the clinic. Every day we're seeing patients with advanced head and neck malignancies who in whom the treatment is incredibly intense and can be debilitating, and in whom, in my opinion, we need better treatments and better approaches to caring for these patients, despite our best efforts thus far. It is the sixth most common cancer worldwide. The majority of these are squamous cell carcinomas that arise on the mucosal lining of the head and neck. They are often related to tobacco and alcohol exposure, although a subset of these are related to human papillomavirus. And an effort in my lab has been focusing on the role of HPV in modulating the therapeutic resistance or the therapeutic sensitivity of these cancers and why these patients have, the HPV-related patients have better outcomes. That's actually not what I'm going to talk about today. The care of these patients is really a multidisciplinary effort. It involves surgeons, radiation oncologists, medical oncologists. Now it includes immunotherapy. And so the care of our patients and our approach to research is really a multidisciplinary one. The overall goal of our lab is improving the lives of our patients by preventing or overcoming therapeutic resistance. Because as I said, although we have much better treatments today than we did when I started training in the field 15 years ago, and better treatments now than we had 25 years ago, we still have a long ways to go. And to illustrate that, these are sort of the average response rates to single agent chemotherapy or multi-agent chemotherapy in patients with metastatic or recurrent head and neck cancer. And clearly, if you have a drug that gives a 50% response rate, that is not good enough, because that means half of the patients who are getting that drug are not going to derive any significant benefit. And so we've, at Wisconsin and in my lab, we've sort of built a lot of our efforts around understanding and studying therapy resistance and how we can improve the results to our current treatments and to the next generation of agents. So I'm going to tell you this morning or this afternoon now about sort of two different stories. The first one is the role of AXL in resistance to cetuximab in squamous cell carcinoma. And this is the project that has been a strong collaboration with the Wheeler Lab. The second is MDM2 inhibition in adenoid cystic carcinoma. This has been largely driven out of our lab and through really some sort of fortuitous findings as we've been developing patient-derived xenografts as a resource for our head and neck cancer spore. But to delve into the first project, so cetuximab, which I imagine many in the audience are familiar with, is a EGFR-targeted monoclonal antibody that has been approved for the treatment of head and neck cancer, both in combination with chemotherapy and in combination with radiation. And although its use has, I would say, waned over the past few years, it is still an agent that is given to a great many patients with head and neck cancer, either as a part of multi-agent chemotherapy, as part of chemoradiation for patients who cannot tolerate cisplatin, or have other contraindications to platinum-based agents, or in the recurrent metastatic setting for patients who can't tolerate multi-agent chemotherapy. And so Derek Wheeler's lab and my lab have spent several years now sort of studying cetuximab resistance and being interested in how cells become resistant to cetuximab and how we can overcome that resistance. And so we started a lot of this work through the work of a really highly motivated, really productive graduate student, Tony Brand, looking at both lung and head and neck cancer cell lines and using a sort of a system in which we exposed cells either in vitro or in vivo to cetuximab. And as you can see, can you see my mouse? Yeah. So as you can see, exposing the cells to cetuximab, although the tumor growth, in this graph is ultimately much slower, these tumors eventually certainly do grow out in what we term a acquired resistance to cetuximab. And I'll talk a little bit later about sort of intrinsic and acquired resistance. And if you look at these different cells, the cetuximab resistant compared to the parental cells, one of the multiple different findings, one of the things that you find is that the level of AXL and phosphoAXL is significantly elevated in the resistant cell lines. And we see that in vitro cultures and in vivo models, shown here looking at AXL expression, both at the protein level and at the RNA level. And so this was some of the initial work that led us down this study. And we see, so if we looked across a panel of patient-derived xenografts, on the left, cetuximab-sensitive models, and on the right, models that are resistant to cetuximab. And the bar graphs here are demonstrating sort of the normalized or the percent tumor growth relative to vehicle treated. So these all are sensitive, and these are all relatively resistant. What you see when you stain from an immunohistochemical standpoint is significantly increased AXL expression in the cetuximab resistant. And these are looking, instead of at the acquired resistance in the last line, at more intrinsic resistance, so without any prior exposure to the EGFR inhibitor, just at baseline, significantly elevated expression of AXL. When we looked in a patient cohort at AXL expression, what we saw was that in patients who have high AXL expression, they have worse progression-free survival than those with low AXL expression. And if you look over from sort of normal oral tissue to poorly differentiated, over that sort of differentiation spectrum, you see that AXL expression increases as you go from the more well-differentiated tumors to the more poorly differentiated tumors. And so, that is all certainly suggestive of AXL playing a role in resistance to cetuximab. So we then went into a couple different cell lines in which we overexpressed AXL or overexpress and then knock down AXL. And what we see is that when you overexpress AXL, shown here, these cells become resistant to cetuximab relative to the vector control. And if you then knock down that AXL expression, they again become sensitive. And the same sort of pattern seen in multiple cell lines. So we looked sort of widely across head and neck cancers, or across our head and neck cancer cell lines at AXL expression, and we see some that express relatively high levels and some in which we can't detect expression at all, and others which it's sort of intermediate. And because my lab had interest in human papillomavirus, one of the things that we often do is think about whether HPV may be playing a role in this. And the cell lines that are boxed in red here are all HPV positive or HPV related head and neck cancers that harbor the human papillomavirus and express E6 and E7. And what you can see is that essentially there's not any clear relationship between AXL expression and HPV status in these sort of established cell lines. So what is AXL? So AXL is a receptor tyrosine kinase. It's part of the TAM family of receptor tyrosine kinases that include Tyrol, AXL, and MER. It has become a very attractive target from a pharmacologic standpoint, with inhibitors targeting AXL, targeting MER and AXL, or targeting all three. AXL itself is activated by the ligand Gas6. MER and Tyrol are activated by protein S. And ultimately, activation of these receptors leads to activation of the sort of standard cohort of intracellular signaling pathways: PI3 kinase, AKT, PLC, GRAB2, SRC family kinases, et cetera. And so sort of going to a lot of the intracellular signaling pathways that many of us have spent time studying. So along the way, in addition to noticing that these sort of high AXL expressing cells, the cetuximab resistant high AXL expressing cells also have elevated levels of HER3 and phospho-HER3 relative to the parental cells, both in a lung cancer model and in a head and neck cancer model, and activation of phospho-Sarc family kinases, phospho-AKT, et cetera. And if we look, so these are the acquired resistance models, and if we look at intrinsically resistant models, we also see that if you block AXL with an antibody targeting AXL, you lose HER3, at least HER3 activation, and you lose Sarc family kinase activation. So this indicated that the Sarc family kinases may be playing a role in how AXL is signaling, in the important signaling downstream of AXL. We looked again in our de novo models, our acquired resistance models, at our de novo models, and at sort of control IgG-treated tumors and cetuximab-treated tumors, and saw that the cetuximab-treated models upregulated phospho-HER3, whereas the control-treated models had sort of lower levels of phospho-HER3. And when we looked in our patient-derived xenografts at the intrinsic models, the cetuximab-sensitive models had relatively low levels of phospho-HER3, whereas the resistant models had, in general, more elevated levels of phospho-HER3. So we then looked at whether overexpression of AXL ultimately leads to Sarc family kinase and HER3 activity. So here, back in our two different head and neck cell line models, so the vector versus AXL overexpressing, and what we see is with strong AXL overexpression, we see both activation of the Sarc family kinases and activation of HER3. And if we use SLAP to downregulate the Sarc family kinases, we lose the overexpression or the activation of HER3, suggesting that the Sarc family kinases truly is playing a role in activating HER3. So then the next step was to look at, so maybe it wasn't just AXL that was important, but is HER3 also driving cetuximab resistance? And so back in our HN4 cells, looking at AXL overexpression or HER3 overexpressing cells, what we see in the cetuximab treatment is that the vector are sensitive, and both with AXL overexpression or HER3 overexpression, those cells are now resistant to the effects of cetuximab. So this sets up sort of the model that AXL overexpression is driving HER3 activation, leading to resistance, and that if you can block HER3, you may resensitize those tumors to treatment. And so we tested that in our AXL overexpressing cell line, in which we used SI-HER3 and then treated with cetuximab, or with or without cetuximab. And so what you see here, just looking at cell proliferation, is that knockdown of HER3 alone had very little effect unless the cells were also treated with cetuximab, in which case cetuximab treatment then resensitized the cells that no longer expressed HER3. So really putting sort of this serial EGFR AXL HER3, where we believe AXL is playing a role in activating HER3 and leading to resistance to cetuximab. And so the area that we're sort of focused on now is understanding, exactly what components of AXL and how AXL is causing this. And so we've now started to go into the tail of AXL, the intracellular tail, and knock out the various tyrosine residues. So here we have the vector control on the left, where these tumors are allowed to grow, and then around 200 cubic millimeters, the mice are treated with cetuximab, and you see, compared to IgG control, those tumors grow quite nicely. The vector cells treated with cetuximab essentially sort of stay the same size over a period of 3 weeks. In the AXL overexpressing cells, you see no effect of cetuximab. And cells that overexpress AXL with a tyrosine 821 mutation are now sensitive again to cetuximab. And we've looked at this in the other phosphorylation sites within AXL, and this 821 is the only one thus far that gives us a robust signal, suggesting that tyrosine 821 is really driving the cetuximab resistance through AXL. And so this is sort of the model that we've ultimately built up, where tumors that are resistant to cetuximab, cetuximab downregulates EGFR signaling, and ultimately that leads to overexpression of AXL, and we have work ongoing to understand better how EGFR loss or downregulation of EGFR, how that is turning on AXL. But AXL functions through Src family kinases, likely through the tyrosine 821, to activate HER3, and that we think probably a combination of HER3 and AXL, sort of the other downstream signaling pathways, ultimately lead to therapeutic escape, proliferation, and survival. And so if we use an AXL-targeted antibody, we lose HER3 overexpression, we lose the activation of these proliferation survival pathways, and ultimately we get increased cell death and resensitization. We and others have shown that if you target HER3, you can get a similar response, and so I think an important question that the field needs to understand is, are we better targeting AXL? Are we better targeting HER3? Are there some of our work is looking at other escape mechanisms now to AXL inhibition, and the other TAM family receptors seem to be playing an important role in resistance to AXL inhibitors. So after doing all of this work, we wanted to look at whether targeting AXL in our mouse models, our PDX models of cetuximab resistance, ultimately resensitizes the tumors to cetuximab, because all the work, or the majority of the work I've shown thus far, is really working in vitro model systems. And so over the past 7 or 8 years, we've developed a robust repository of patient-derived xenografts, and one of the things we've done, at least in a cohort of them, is characterize their response to cetuximab. And here shown just graphically as the tumor growth curves, showing both sensitive and resistant models, and here quantifying tumor size relative to control. And so the next slide is going to show you data using SCC1, which essentially has no response to cetuximab, and SCC64, which is modestly responsive to cetuximab, at least in that initial study. And so here we're treating these tumors with vehicle, with cetuximab, with R428, which is a small molecule inhibitor of AXL, or with R428 and cetuximab. And so in our SCC1 model, sort of the de novo resistant model, we see again cetuximab itself in the triangles has very little effect on tumor growth compared to control. AXL inhibitor by itself has very little effect, but the combination therapy has significant growth delay. In our SCC64 model, which we chose because we expected it to be at least partially responsive to cetuximab, this is now a later generation of that model. I believe this was passage 5 and the previous was passage 3. We now see that this model at passage 5 is actually not sensitive at all to cetuximab, but the combination treatment, again, is significantly better than either treatment by itself. And so this sort of illustrates sort of two different points. One, that these patient-derived xenograft models certainly can evolve over multiple passages, and that to just have done an experiment using combination treatments and not to have repeated our controls would have potentially led to very different interpretation of some of the data. And so sort of a word of caution, as I know Antonio and others here know, these things are living, evolving model systems, and so we have to be careful as we utilize them. So this project, as I've sort of described, is Project 4, as it was the fourth project brought into our head and neck cancer SPORE grant. And as part of that project, the last aim is a window of opportunity trial in which our goal is really to confirm that this mechanism of resistance via upregulation of AXL and HER3 that we see in all of our preclinical model systems is actually occurring in patients who are receiving cetuximab. And so this is a very simple study in which patients are going to have a research biopsy. Patients who are scheduled to undergo surgery will have a research biopsy. We'll be collecting circulating tumor cells, and I'll talk in a moment a little bit more about that. That research biopsy will be scored for AXL expression, for HER3 expression, for Ki67, and used for PDX generation. And then the patient will receive 2 cycles of cetuximab, or 2 doses of cetuximab, and then go to surgery. And we'll compare the surgical specimen to the pretreatment biopsy specimen. We'll repeat circulating tumor cell collection at that point. At 1 month, they'll come back for a post-op visit to discuss possible adjuvant therapy, and we'll do another CTC collection. And in this, with this trial, we'll be able to do 2 things. We'll, one, be able to see whether patients who have a response to cetuximab have upregulation of AXL expression, and whether the PDXs in which we know how the patient responded, whether the tumor in mouse responds the same way the patient does, or is there a discrepancy between how the mouse responds and how the patient responds. And currently, although we know how our PDXs respond, because most of those came from surgical specimens, we really don't understand how the patient would have responded to that drug, because most of them had surgery either long before they received any chemotherapy, or there was such an interval between chemotherapy and surgery that having a firm understanding of how the patient responded has been impossible. So we've, as other groups have done, we're using Ki67 as our surrogate marker of response, although we'll be doing tumor measurements. And in our preclinical systems, if you look at cetuximab sensitive versus cetuximab resistant xenografts, you can see that the cetuximab sensitive have a significant drop in Ki67, and the cetuximab resistant have very little effect on Ki67 due to the effects of cetuximab in blocking proliferation of these tumors. And this, as a surrogate marker, has been used by several other groups looking at cetuximab, and as a quick and easy way so that even if we don't see a big effect from a tumor volume reduction, we still expect to see an effect from a proliferation standpoint. And we see this actually when we've looked in our model systems. After only a day or 2 of cetuximab treatment, we can see a big drop in the proliferation index. And so that's our study has been powered based on the expectation of the change in Ki67. And as I said, we'll be identifying or generating PDXs in which we know the patient was resistant to cetuximab, and we'll take those and test whether targeting AXL in those in combination with cetuximab leads to an improved response compared to cetuximab alone or the AXL inhibitor alone, setting up the data, in our opinion, to support sort of the next generation of this trial, in which we would actually look at combination treatments and seeing whether we can generate better responses in patients treated with an AXL inhibitor and cetuximab compared to just cetuximab by itself. Our CTC collection has been the evolution of a collaboration with Sung-Pao Hong, who is a faculty in the School of Pharmacy. And Sung-Pao has been at Wisconsin for about 3 years and was previously in Chicago, where he and his colleagues developed a circulating tumor cell nano sort of captured dendrimer-based assay in which they utilize sort of a slow-rolling E-selectin-coded region patterned with their antibody, their CTC capturing regions to increase the collection of circulating tumor cells compared to just an antibody-coated membrane alone. In addition, they've done a lot of work looking at these sort of nanoparticles and the importance of the size of the nanoparticle in capturing more circulating tumor cells. And we are actually, through a developmental research grant through the SPORE, working at optimizing this CTC assay specifically for head and neck cancers as opposed to just an all epithelial cancer CTC collection. And so here's a little bit of their data from their recent publication looking at the benefit of adding these E-selectin coded regions to slow down the capture of the cells. And you can see the purity of the cell capture goes up significantly with the E-selectin coding. And they've done some initial characterization or comparison to another commercial product and shown significantly more CTCs that they can detect per mil than the other commercial project. This is now a company that I have zero intellectual or financial interest in, but that Sung Pao is certainly involved in. And so didn't show up on my disclosures, but ought to at least mention that. And so they worked with some of our friends at the University of North Carolina, someone he had been postdocs with in Boston, and looked at patients treated with radiation pre-treatment and post-treatment, showing that their assay is capable of detecting changes in CTC counts pre and post-treatment. And here just showing an individual patient who was followed from pre-treatment levels, mid-treatment, end of treatment, 1 month post-treatment, and then this is sort of a rise 6 months post-treatment presaging a clinical recurrence in that particular patient. They have not yet looked in patients who've had their disease resected, and so that's one of the things that we're going to be doing is hoping to see a significant difference in the CTC numbers post-surgery, at least in those patients who have what we believe is a complete resection, and to sort of show that aspect of it is really sensitive to significant changes in tumor bulk. So sort of to summarize this part of the talk, cetuximab resistance in head and neck cancer is mediated at least in part through AXL upregulation. We believe that occurs, that AXL upregulation ultimately activates HER3, and that if you block at either of those points, you can sensitize head and neck cancer to cetuximab. And we are hopefully this week, if not this week, next week, opening this window of opportunity trial. It's passed the IRB. The NIDCR has finally signed off, and we're just in sort of the final hospital approvals to open this trial, which we're hoping to enroll about 30 patients over the next 18 months in order to confirm the mechanism in humans and to lead into the next clinical study. So that is sort of the end of the first story, and I'll turn now to the second story. And the second story relates to salivary gland cancers and adenoid cystic carcinoma. Salivary gland cancers represent about 3% of all head and neck cancers, and adenoid cystic carcinomas are about 10% of these salivary gland tumors. Salivary gland cancers, and in particular adenoid cystic carcinoma, have essentially no highly active agents to treat these patients, and best reported response rates range between 20 and 40% with either single or multi-drug regimens. Importantly, and you'll see why, the genome exome sequencing studies in adenoid cystic carcinoma have identified a very low rate of P53 mutations, and that sort of led to some of our initial studies, some of which have been done sort of in parallel by Jacques Noor at Michigan, who has developed some adenoid cystic carcinoma models, as well as Antonio and colleagues here. So it so happened that one of the relatively early patient-derived xenografts that we developed at a point where we were trying to get fresh tissue from any patient having surgery was from a patient who had lived for 20-some years with metastatic adenoid cystic carcinoma, but who was having probably his fifth or sixth operation to remove locally progressive disease. And this adenoid cystic carcinoma, so I show here H&E and some immunohistochemical markers comparing the primary, the first passage, and the fifth passage, had good stability in our hands, at least from a IHC standpoint over multiple passages. We did selected gene mutation or gene panel mutation analysis, identified a previously undescribed VEGFR and KIT mutation, and importantly for this story, had very good coverage of P53 and showed P53 to be wild type in this particular patient. And as I said, adenoid cystic carcinoma has P53 mutations as indicated by this red arrow in about 5% of tumors, so much lower than sort of your average rate of mutation in P53. And Jacques Noor's group had published, sort of just as we were starting this work, evidence that using an MDM2 inhibitor, a compound that they had developed in-house at Michigan, showed some growth delay in their model system of adenoid cystic carcinoma and some degree of synergy with cisplatin in their model systems. So we were interested, as a radiation oncologist, we were interested in combining or in looking at drugs combined with radiation. And so we utilized AMG 232, an Amgen compound which sort of inhibits MDM2 and blocks its interaction with P53 with the idea that radiation is a strong inducer of P53. And that if you can block the MDM2 inhibition of P53, you can perhaps sort of supersize, if you will, the P53 response, leading to cell cycle arrest, and hopefully not so much the functional DNA repair, but more the apoptosis route. And so we had, Paul Harari's group had some initial data in P53 wild-type lung cancer and head and neck cancer, and as we developed this model system, we felt like this would be a good system in which to look at this. And so because we didn't have any, because at this time there were 0 adenoid cystic carcinoma cell lines available worldwide, that has since changed both through work here and at Michigan. We did all of this work in vivo. And so here I'm going to sort of sequentially show the tumor growth curves. And as you can see, these are relatively slow-growing tumors. This is a control-treated tumor. Chemotherapy and all of these will be delivered from around day 17 through around day 41. And radiation given at 5 gray per fraction in this initial pilot study. And what we saw was that radiation alone led to really, at relatively high doses, 40 gray, 5 gray per fraction twice a week, led to really good stability. If you followed these tumors out long enough, they eventually did regrow. And so we didn't expect a whole lot out of the next 2 slides that I'll show you. So the AMG alone actually led to pretty good tumor control that during the course of treatment and then after treatment stopped, the tumors started to grow again, not very unexpectedly, given the fact that we didn't really see regression of the tumors. And what was remarkable was that when we combined it with radiation, I had to change the scale to a log scale because the tumors just disappeared, totally gone, and did not come back. No matter, we followed these out to around 180 days. So a really remarkable response that I was a little, we don't see this type of response in our preclinical model systems very often. Sana and David can attest to that. Even though radiation is pretty darn good at killing cancer, this was pretty remarkable, and I was a little highly skeptical that this would be repeated. And so we went back to repeat it, and this time, because we saw such a big response, we wanted to see if we could titrate that response a little. So we changed our radiation regimen and used 3 different doses of radiation. Prashant Prabhakaran and Amal Javad, who did a lot of this work, they were juggling a tremendous number of animals with really complex sort of schedules and schemas in terms of treatment. So here, for simplicity's sake, is the control-treated tumor on the top, and we see this nice dose response, 2 gray, 5 gray, 8 gray. But essentially, although 8 gray times 5 or times 8 did result in some tumor regression, after treatment ended, these tumors certainly regrew. These were all flank models, not orthotopic systems. And again, the MDM2 inhibitor, AMG 232, resulted in, again, a modest slowing. There was some growth during the treatment that sort of accelerated to some degree afterwards. That's a lot of radiation to some of these, dude, if that's what you're looking at, counting the radiation fractions. It's a lot of radiation we gave. We wanted to see if we could eliminate the cancer with just the radiation alone, and we did not. And here, what you can see is when we combined the radiation, the exact same radiation that I showed initially above, when we combined that with the AMG 232, what we saw was this really remarkable, not just tumor slowing, but even with 2 gray times 8, this regression during the course of treatment, and then continued regression afterwards. And so the dotted blue line is 2 gray, the dashed blue line is 5 gray, and the dots and dashes are 8 gray. And all 3 of them, if you combine the drug with radiation, the tumors went to nothing. Like no palpable tumor, couldn't feel it, dissected, couldn't find anything under there. And here, sort of shown graphically, so here are, selected tumors, of course, but, no radiation, large tumors on the flanks of the mice, AMG, smaller, but still certainly tumors, 2 gray, 5 gray, 8 gray, radiation alone on the top, radiation with drug on the bottom, and there's just nothing there. The mice are, relatively fat and happy and living okay. They had some, issues with diarrhea and weight loss during the initial radiation delivery, but recovered pretty well afterwards. And so, I had not had an experiment where before this, that we had ever thought of calculating a TCD50, a tumor control dose, where you control 50% of the tumors, and essentially, 8 gray, 5 gray, 2 gray, we were all above 50%. So this is just all modeling, and given the number of animals and the cost of this, we didn't want to go out to lower radiation doses. It just didn't seem to make any sense. But, compared to the control with radiation alone, where you didn't get control of any of the tumors, the response with radiation and drug was really pretty remarkable. So we took those tumors because, again, we didn't have any in vivo or in vitro model systems, and we looked at P53, and so what you see here, the radiation alone, you see a little bit of P53 activation with the higher doses of radiation. All of these tumors were harvested 24 hours after the initial dose of radiation or initial dose of radiation and drug. And in the AMG-treated tumors, you see more P53 without radiation, but much stronger induction of P53 with radiation, and likewise much stronger activation of P53 when looking at phospho-P53, suggesting that this is a specific effect of the drug hitting its intended target and leading to functional P53. If we looked downstream at P21 or CDKN1, both at an RNA level or at IHC, we again see this strong dose-dependent, AMG-dependent induction of these P53 downstream target genes, suggesting that, again, this treatment, in particular the radiation, is really inducing this P53 response in these P53 wild-type tumors. And if we looked at proliferation in these, we saw more modest effects. These are, as you saw from the growth curves, relatively slow-growing tumors, and so the Ki-67 index is awfully low to begin with, but radiation alone had minor, if best, effects on the number of cells that stained for Ki-67, but when combined with AMG, we saw a significant, at the highest doses, a significant elimination of the Ki-67 staining in those cells. And when we looked at apoptosis via cleaved caspase or calculating the number of, here looking at cleaved caspase, we see this strong induction of apoptosis via cleaved caspase 3 staining in the AMG plus radiation compared to AMG or radiation alone. And if we counted apoptotic bodies, or if our pathologist counted apoptotic bodies, we saw, again, a strong induction of apoptotic bodies per field in the combination treatment compared to the individual treatments, particularly at the higher doses of radiation, where we saw the fastest tumor response. So, beautiful, in my opinion, really beautiful data. We have been relatively unsuccessful thus far in obtaining, either generating additional models. We've put additional adenoid cystics in and haven't had great success in growing them out. We are waiting on MTAs from Michigan to get their cell line, their published cell line, and some of their adenoid cystic carcinoma models in-house to retest these. And with the help of the Adenoid Cystic Carcinoma Research Foundation, we have been talking with companies. There are numerous companies who have MDM2 inhibitors. We've been talking with some of them about the potential of a clinical trial in adenoid cystic carcinoma of an MDM2 inhibitor combined with radiation. And thus far, we've had no real success in generating anything other than sort of initial interest in discussions. So that leads me to sort of the last couple of slides, which is that about 5 months ago, one of our colleagues across the street at the School of Pharmacy called me up because he had come across our paper when he did a PubMed search of MDM2 and Wisconsin, and called up and said, hey, I'm in School of Pharmacy. I'm interested in developing PROTACs, proteolysis targeted chimeras, which are these drugs, sort of these two-part drugs, one of which is specific to a protein of interest that you want to degrade, the other of which is specific to ubiquitin ligase, and where there's a small linker in between. They can change the ligase, change the linker to ultimately lead to the ubiquitination of our protein of interest and degradation. So they have been developing these PROTACs over the past couple of years and have developed an MDM2 targeted PROTAC that they had tested in one cell line when his latest postdoc recruit's wife was a cell biologist and looking for something to do. She came in and volunteered in the lab and ran some tissue culture experiments, but they really hadn't done, they were a protein interaction chemistry lab, and so they hadn't done a lot of biology or cell biology. And so we got this in our hands right around Christmas time and have started to look here in a P53 wild type cell line, initially just at drug alone, and we see at least some response to drug alone. My technician this week is looking at this drug in combination with radiation in clonogenic survival assays. I was hoping I'd have some of that data to show you today. And ultimately, our plan is to take this drug into our adenoid cystic carcinoma model and see whether we see the same effects in our model system with the idea that this is now an in-house compound that Wisconsin is in the process of developing and that we may be able to bring into a clinical trial on our own, taking out the drug companies in some of those discussions. And so we're sort of starting some of those discussions. They've talked with WARF, our intellectual property group, because this is all sort of in their field of interest. But I think it's another example of the types of collaborations that can develop when there's really great people, or when you can surround yourself with really great people, which is one of the things that I've been very fortunate to have been able to do at Wisconsin. So we've developed, in summary for this part, we've developed this robust model of adenoid cystic carcinoma, although just an in vivo model. And this is a disease where there are relatively few tools out there to study it. And the more tools that our various groups can develop, the more chances we're going to have of finding better treatments for these patients, many of whom can live for decades after diagnosis of metastatic disease, but still, ultimately most of these patients develop symptomatic disease that we need better treatments for. In our system, the addition of AMG 232, an MDM2 inhibitor to radiation, greatly improved response, leading to durable tumor regression and what I would call complete responses, really in a way that was quite remarkable compared to any other model system that I've studied over the past 20 years. And this combination therapy, as we sort of expected, led to P53 activation, inhibition of cell proliferation, and activation of apoptosis. So again, this work that I've showed today is the work of a lot of different people. I'm just one small part of that. The Axel work has been a really fun collaboration between Derek Wheeler's lab and my lab. Tony Brand was the initial grad student, and now Nellie McDaniel is the current grad student focusing on that project. Rong is a pathologist who's been a tremendous asset to us over the past 4 years since she came back to Wisconsin. Emmanuel and Irene involved in some of the statistical and sequencing data that I showed you. Sung Pao, the CTCs, Wei-Ping Tang, the MDM2 PROTAC. Of course, the people in my lab. The work that I showed from my lab today was the work of Adam Swick, the postdoc who sort of led 2 post-bac students, Amal and Prashant, through that work. And then a lot of other people over the years, a lot of undergrads. We're fortunate at Wisconsin to have a huge undergraduate population with a number of students who are really highly motivated. And we've been lucky to have a number of them who've stayed in the lab for post-bac years after spending a few years initially, and really had great productivity from them. And of course, the entire head and neck team that Paul and Greg Hardig have built over the past 20 plus years at Wisconsin, ultimately culminating in our head and neck cancer SPORE, which is 2.5 years in. And we're now in the process of talking about the renewal and projects into the renewal. And then funding, although none of this work was partially supported by the Pharma Foundation through a postdoc fellowship to Adam Swick. The lab has funding from the American Cancer Society, the V Foundation, and the NIH, which weren't necessarily related to the work that I showed here, but certainly are out there. So happy to take any questions. I think we have just a couple more minutes.
With your MDM2 have you tried combining it with a systemic agent? Do you think that the tolerability of that could be possible with a systemic attack?
So we have not tried combining with systemic agents, largely because Jacques Nora's group at Michigan had done that with cisplatin with a different compound, but an MDM2 inhibitor, and seen, at least in their preclinical models, reasonable control, although not the degree of tumor regression that we saw combined with radiation. And I believe they have a clinical trial now going, looking at the combination of the MDM2 inhibitor and cisplatin in patients. I'm not sure if that's open, but that's certainly in their plans. Yes.
AMG inhibitor, have you considered looking at system where you have an intact immune system,
So that's a great question. So I didn't show any of our humanized model work, but like the group here, we've been working with a transplant surgeon at Wisconsin who has developed a humanized model that involves thymic fragment and cord blood reconstitution. That model is not yet at the point where we feel confident in using it in that type of system. But I think that's a really good question, and whether that sort of robust apoptotic response can elucidate a stronger immune response. David?
So understanding how adjuvant system is typically treated in the clinic, and I don't know how it is in Wisconsin, but you know that cancer typically is attacked surgically, and we're asked to offer post-op, and that's what the RTOG studies are.
Yep.
And we also know that in history related, it's kind of like radiation related. It's the gift that keeps on giving.
Yeah.
And 5 to 7 to 10 years out is when patients start showing up with lung mets.
Yep.
In that context, what's your vision then of a clinical trial? Over the last 25 years, we've shown improvement over control, and yet that doesn't seem to alter the different metastatic pattern. How would you utilize
Right.
what you're doing?
So what we've talked about as our first step is looking in, I think like you, we often see patients who have local recurrences. And so using them as our initial step, where we can take a patient who has a local recurrence, many of them have had, or most of them have had radiation before. And so now we're talking about reirradiation. And given our robust responses with, 2 gray times 8, which is really little dose, doing a trial where we, sort of either slowly de-escalate the radiation dose, looking at that local response, or started a really low dose in combination with drug and increase the radiation dose until we see good response, looking at, tumor responses in the local setting, because giving, radiation to, usually when these patients develop lung mets, it's not one lung met, it's 30 lung mets. And so that to me is not a great place to go unless we're giving 2 gray, a single fraction of 2 gray or something like that. But I think without better, more robust data in patients, I'd feel more comfortable doing it in that reirradiation setting or unresectable or in a pre-op setting, right, to give, drug and 10 gray pre-op, something like that would be the other possibility. I think the pre-op setting is a great idea. I saw another hand up somewhere, I thought. No? Okay. Oh, Antonio.
CTCs, it's a scary slide. Our patients have 35 CTCs per ml. Right?
Yeah.
However, head and neck distant metastases are relatively, fortunately, they're relatively uncommon.
Yep.
So, do you see those cells as being real active cells, or are those the ineffective cells that are not going anywhere? And how concerned are you as using them as surrogates for your IIT?
So I think that's a really good question, and that's so. They will. So in the SPORE trial, they are not the primary or secondary. They're a tertiary endpoint, sort of an exploratory endpoint. So I think from that standpoint, and the other component of that is that these patients are going to be having surgery, although we're including patients who are having surgery, for recurrent disease, and maybe they have metastatic disease, we're including those patients. The majority of the patients we expect are going to be having definitive surgery, and so we anticipate those CTCs to go to undetectable levels. They've been doing one of the areas they've been spending a lot of time lately is, taking normal donor blood and trying to refine their surface so that they capture, 1 cell or 2 cells in normal donor blood, and defining what is the level that is positive versus negative. Because with these assays, you're certainly going to detect you're capturing epithelial cells, you will detect some cells, but they see with sort of the dual antibody approach, they see relatively low numbers, like in the low single digits per mil in normal healthy donors. So, great, thank you.